The dark band is made up of highly refractile material which is called as A band, that is anisotropic band, which is having a length of 1.5 microns. In the center of each A band is found a slight less refractile region which is called as H band. M line is seen in the middle of H band due to the central bulge of A band. The alternate light band is made up of lower refractile material, therefore it looks lighter and is called as I band, that is isotropic band, which is having a length of 1 micrometer. In the center of each I band is found a narrow line, highly refractile material which looks dark, called Z line. The contractile unit of the muscle is the substance between two adjacent Z line, which is called as sarcomere and having a length of 2.5 microns. This is a diagram showing a sarcoplasm. Sarcomere is the area between two Z lines, and each sarcomere, they are made up of myosin bands which are thick and actin filaments which are thin, and so they have been divided into dark band and light bands. Dark band is A band, whereas I band, the light band is I band, which is anisotropic. Electron microscopic view. The myofibrils are made up of two sets of protein filaments, thick and thin filaments. Thick filament, it is made up of myosin, which is responsible for the formation of A band. Myosin molecule is made up of 6 polypeptide, 2 heavy chains and 4 light chain. Myosin treated with proteolytic enzyme, it reveals 2 fragments. Short compact head of meromycin, which is called as HMM, it has 2 important sites, that is actin binding site where myosin comes in contact with actin, and there is the ATPase site that hydrolyzes the ATP. Long chain of light meromycin, it has only structural function and it acts as a backbone for HMM. This is the structure of the myosin, where it has got the heavy chains and the light chains, 4 light chains, and this is divided into head part and the tail part, and head contains the actin binding site, and it also has a site for ATPase activity. Thin filament, it is made up of actin, tropomyosin and troponin. They arrange themselves to form 2 chains of globular units that form double helix, which is responsible for I band. Each thin filament contains 300 to 400 actin molecules and 40 to 60 tropomyosin molecule actin. It occurs in 2 forms. The actin occurs in 2 forms: G actin, which each molecule bind with 1 molecule of ATP firmly, and F actin is formed by the polymerization of the G actin. Tropomyosin, it covers the binding site of actin where myosin head comes in contact with actin, that is, it prevents the interaction between actin and myosin. Troponin, they are small globular proteins made up of 3 subunits. Troponin T, which binds the other troponin components to tropomyosin, whereas troponin I inhibits the interaction of myosin with actin, and troponin C contains binding site for calcium that initiates